In this video, I'm going to tell the viewers about the feed fast cycle. And before going into in-depth discussion of this topic, I want to make it clear that all of those who have problem in understanding the concepts of biochemistry metabolism, it is very useful to start your biochemistry by understanding the concepts of feed fast cycle, because the problem is that when we start with metabolism, the concepts are given us in separate forms. Means that for example, if you have glycolysis, you understand glycolysis pathway, you understand its regulation. Similarly, then you move to gluconeogenesis, and then you move to lipid, you move to protein. In that way, the concept is given in separate parts, and it is very, because being a volatile subject, biochemistry, you forget that concept when you move to next topic, and you are not able to link those things if you have not read it so closely. What feed fast cycle does is that it unites all those concepts that you need to know at one time, so that you have a map of what is happening in your body metabolism. You have a clear cut division that this is going to happen with this substrate, this is going to happen with this substrate. After studying feed fast cycle, what is remaining is this: just you need to memorize the pathways and a little bit of other concepts, but the whole concept of the biochemistry metabolism in this single topic. So I would recommend everyone to start your revision of the biochemistry with, or whatever you start biochemistry with, feed fast cycle understanding, because that is the thing that will lead you to a mastery level. Okay. So now, after understanding the importance of feed fast cycle, I need to tell you about two important hormones that are going to play a very crucial role in understanding the biochemistry. The one is insulin and other is your glucagon. These two hormones play a crucial role. I mean to say that one is insulin and other are other hormones which are having which show anti-insulin effect or counter regulate the insulin. These are listed as glucagon, epinephrine, norepinephrine, your growth hormone. Okay, these hormones counter regulate the insulin, and the importance of understanding these two mechanisms is that insulin is the predominant hormone that acts in your fed state, when you are completely fed, when you are in absorptive state, you have just eaten your food. Insulin is the dominant hormone that acts in that phase. And when you are in fasting phase, in between your feeding phases, the predominant hormone that is active is glucagon. So you have to clearly born in your mind that in feeding state the predominant hormone is insulin, and in fasting state it is glucagon. We can also consider this by saying that the insulin to glucagon ratio is high during the feeding phase. Feeding phase is also known by the name of absorptive phase, and the insulin glucagon ratio is low in case of your fasting phase. So before understanding the concepts of feed fast cycle, you need to understand that the insulin and glucagon, how do they act and how do they influence the enzymes, which are the enzymes which are going to take part in the pathways of biochemistry. And that is where your concept building starts. So first, in this I would start with insulin. What you're seeing in this particular image is this is the receptor of insulin. This is a receptor of insulin. These are two alpha subunits, these are two beta subunits. Now you can clearly see that the beta subunits are spanning the membrane, so they are transmembrane. And what you see, this alpha which is united with beta via disulfide bridges. Now that is not important what I just told, even if you forget this. You need to remember this thing, that only that insulin binds to alpha subunits. This is the reason where your insulin is going to sit. This is the reason where insulin is going to sit and bind, and that's where the insulin comes. It binds to your this particular alpha subunits. Okay, and ultimately what it does basically, I need to tell you about that beta subunit is special in one thing. Why? Because beta subunit has tyrosine residues attached to it, and these tyrosine residues when insulin binds to alpha subunits, these tyrosine residues will be autophosphorylated. Phosphorylation means attaching a phosphate molecule. They will be autophosphorylated with this, and this causes the insulin receptor substrate. Are the these are the insulin receptor substrates. These receptor substrate will be phosphorylated. This is a autophosphorylation. This is a property of autophosphorylation, that it gets phosphorylated on itself, and this will cause the phosphorylation of insulin receptor substrates, which after becoming phosphorylation, after undergoing phosphorylation, what insulin receptor substrate does is it will cause, देखो होता क्या है कि phosphorylation is going to continue. I mean, this gets phosphorylated, this will continue phosphorylating IRS phosphorylates other enzymes. Now these enzymes are predominantly two. Either it is phosphatase, or either it is what? Phosphorylase or kinase enzyme. So there are two enzymes that we need to know: the kinase and phosphatase. This is your kinase enzyme and this is your phosphatase enzyme. So what insulin is going to do, these IRS which are phosphorylated is either going to phosphorylate kinase or either going to phosphorylate phosphatase. Okay, and if it is phosphorylating kinase, it is activating kinase, and if it is phosphorylating phosphatase, it is activating phosphatase. And with this, what happens? Once these kinases or phosphatases are active, they are going to play their role, because kinase in itself causes phosphorylation and phosphatase causes in itself dephosphorylation. So that is how the biological effects of insulin will come. Now, important point is that I just told you that due to binding of insulin to insulin receptor, either kinase is going to act or phosphatase is going to act. Okay, but remember this thing. You need to know that insulin activates both kinases and phosphatase, but remember this one thing, that even if you forget this and you remember only thing that insulin is going to activate only phosphatase, there will be no problem. Why? Because a predominant enzyme that insulin activates is phosphatase, and that is why after activating phosphatase, insulin causes dephosphorylation of many enzymes in the pathways. And when these enzymes are dephosphorylated, these enzymes got active. Okay, these enzymes got active. Most of the enzymes that are dephosphorylated by insulin are active. They become active, and when they become active, they undergo their function. But there are exceptions to this rule also, that insulin causes dephosphorylation. You forget everything that I just told you. I just told you in detail that there's a autophosphorylation of tyrosine residue which causes phosphorylation of IRS, and IRS causes what? Activation of kinases or phosphatases. Now predominantly it causes activation of phosphatases, remember this thing. And when it causes activation of phosphatases, so this phosphatase is going to undergo dephosphorylation of various enzymes, and that is how the biosynthetic effects of insulin will come. So it is what we need to know. The bottom line is that the insulin is a dephosphorylating enzyme. It dephosphorylates the enzymes it act upon, and after dephosphorylating the enzymes it act upon, it causes their activation. But my friends, there are three enzymes which are exceptions of this rule. What are these enzymes? There are three enzymes. Two enzymes take part in glycogenolysis, and one enzyme is hormone sensitive lipase. The two enzymes of glycogenolysis is glycogen phosphorylase, and second enzyme is glycogen phosphorylase kinase, and third enzyme just I told you is hormone sensitive lipase. These three enzymes, when they get dephosphorylated by insulin, they become what? They become inactive. All other enzymes which are dephosphorylated by insulin, they become active. Okay, these are the enzymes, when they become dephosphorylated, they become inactive. Okay, so this is what I am going to what I was telling, that in fed state most of the covalently modified enzymes are in dephosphorylated forms and are active. There are three exceptions. One is glycogen phosphorylase kinase, glycogen phosphorylase, and hormone sensitive lipase. These two above two enzymes are the enzymes of glycogenolysis. They and one is hormone sensitive lipase. What are their functions? I'll just tell in few minutes. But what we need to remember here is that these enzymes got inactive on dephosphorylation. Okay, and remember this thing, it is a vice versa kind of situation, that if an enzyme is inactive in its dephosphorylated state, it will be active in its phosphorylated state. Similarly, if an enzyme is active in its dephosphorylated state, it will become inactive in its phosphorylated state. Now, why I'm telling you is this? Because why I'm telling you this is because if you understand the same mechanism that how glucagon acts in the cell, you will understand this concept. This is the glucagon and this is the cell membrane on which the glucagon receptor is this. This receptor is actually a G protein coupled receptor. What glucagon does is it activates, after binding glucagon to its receptor, the adenylyl cyclase is activated. Adenylyl cyclase converts ATP to CMP and CMP activates protein kinase A. Remember this thing, this enzyme is important. This is protein kinase A. This protein kinase A is activated and this protein kinase is a kinase. So it will cause phosphorylation of enzymes. Now remember this thing. Here it is written that this phosphorylation of enzymes could either lead to activation of those enzymes or it could lead to inhibition of enzymes. Okay, so after understanding the concept that how glucagon acts, you can compare it with insulin, that insulin causes predominantly dephosphorylation and glucagon causes predominantly phosphorylation. You have to just cram this thing right now, that insulin causes dephosphorylation of enzymes and glucagon causes phosphorylation of enzymes. And when insulin causes dephosphorylation of enzymes, it makes the enzymes, most of those enzymes, active, because most of the enzymes they are active in their dephosphorylated state. Exceptions were three enzymes just I told you: glycogen phosphorylase, glycogen phosphorylase kinase, and hormone sensitive lipase. And glucagon, after causing phosphorylation of enzymes, it mostly causes inhibition of the enzymes. But remember these things, the exceptions of insulin will become, for example, the exceptions were those three enzymes which were inactive on dephosphorylation by insulin. Now when glucagon causes them to phosphorylate, they will become active. So clearly you understand the concept that any enzyme which is dephosphorylated by insulin and it becomes active, it will be inactive when it will be phosphorylated by glucagon. Similarly, any enzyme which will be inactive on phosphorylation by glucagon, it will be active on dephosphorylation by insulin. Simple concept.